Even if you assume we have a whole mouse brain connectome, I would hazard a guess that we would have a problem or a challenge to read out any memories from it a priori. And I think maybe that is the frame we can think about this for one moment. Like, even if we have the whole connectome, can we read out a memory from it?
If you want to decode a non-trivial memory, then one of the things you want to do is to do more than just identify this animal has learned something, and here we can find what it has learned. But you want to be able to differentiate between different memories.
We have models of how these systems work. These are model organisms that have had decades of research on them. And so the very first thing that you're doing is you're saying, here is our model of the hippocampus. Here is our model of how place cells and grid cells interact with each other. Now, given that model, which could be wrong, what is the experiment that we could do to try to decode the memory based on that model?
Okay, I'm going to ask the question that I think needs to be asked, which is, non-trivial is doing a lot of work here. What does non-trivial mean? And actually, I would love if some folks, came up to the microphone and offered their perspective on what does non-trivial look like.
This is Brain Inspired, powered by The Transmitter. Can you look at all the synaptic connections of a brain and tell me one non-trivial memory from the organism that has that brain? If so, you shall win the $100,000 prize from the Aspirational Neuroscience group. I was recently invited for a second time to chair a panel of experts to discuss that question and all the issues around that question. How to decode a non-trivial memory from a static map of synaptic connectivity. Before I play that recording, let me set the stage a bit more here. Aspirational Neuroscience is a community of neuroscientists run by Kenneth Hayworth, with the goal from their website to, quote, balance aspirational thinking with respect to the long-term implications of a successful neuroscience with practical realism about our current state of ignorance and knowledge, end quote. One of those aspirations is to decode things. Memories, learned behaviors, and so on. Decode those things from static connectomes. Aspirational Neuroscience holds satellite events at the Society for Neuroscience annual conference, and they invite experts in connectomics from academia and from industry to share their thoughts and progress that might advance that goal. In this panel discussion, we touch on multiple relevant topics. One question is what the right experimental design is, or designs are, that would answer whether we are decoding memory. What is a benchmark in various model organisms and for various theoretical frameworks? We discuss some of the obstacles in the way, both technologically and conceptually, like the fact that proofreading the connections in a connectome, manually verifying and editing those connections, is a giant bottleneck, or like the very definition of memory. What counts as a memory, let alone a quote-unquote non-trivial memory? This year there were 5 panelists, including Michał Januszewski, who is a research scientist with Google Research and is an expert in automated neural tracing. Sven Dorkenwald, who is a research fellow at the Allen Institute and was very involved in the first full Drosophila connectome paper that happened recently. Helene Schmidt, who's group leader at Ernst Strungman Institute. She's an electron microscopy expert and deals with the hippocampus connectome. Andrew Payne, who is the founder of E11 Bio, which is a focused research organization. And he is an expert in expansion microscopy and viral tracing. And finally, Randall Kuhn, who's the founder of Carbon Copies Foundation. Randall's a computational neuroscientist dedicated to the problem of brain emulation. These people take lots of questions from the audience once I do my best trying to get the conversation going with a few questions. I do apologize that the audio is not crystal clear in this recording. I did my best to clean it up, and I take full blame for not setting up my audio recorder to capture the best sound. So If you're a listener, I would encourage you to check out the video version, which also has subtitles throughout for when the language isn't clear. And mostly those subtitles are correct, but if you're a keen observer, you will note when they are not. Anyway, this is a fun and smart group of people, and I look forward to another one next year, I hope. The last time I did this was episode 180. BI 180, which I linked to in the show notes. It was another panel discussion like the one I'm about to play for you. And before that, I had Kenneth Hayworth on, whom I mentioned runs Aspirational Neuroscience. I had Ken on with Randall Kuhnen, who was on the panel this time. They were on at that time to talk about the future possibility of uploading minds to computers based on connectomes. So that was episode 103, which I also linked to in the show notes. And those show notes are at braininspired.co/podcast/227. All right. I hope you enjoy this panel discussion. As you'll see, there are plenty of issues to resolve, plenty of optimism. I'm the only pessimistic person there that voiced that opinion anyway. So that was kind of interesting, but lots of interesting discussion. Okay. I hope you enjoy it.
Hi, so I'm Randall Kuhnen, and I run a nonprofit research foundation called the Carbon Copies Foundation. I used to work at a company called Voxa, working on the high throughput electron microscopy pipeline, doing the software side of that. They were contracted to the Allen Institute, working obviously on their big project. And in a previous life, I used to model hippocampal, entorhinal, episodic memory systems, and also prefrontal cortex, TD learning, like reinforcement learning sort of phenomena that you could find there. And now my focus is on trying to validate or ground truth the transition from structure to function, so that mapping and building an architecture, deciding how to estimate your parameters, and how can you tell that what you're ending up with is actually mapping towards most of what the tissue that you have data from is actually doing and cares about, has some sort of meaning. So the memory decoding prize is super interesting to me for that reason.
Hi, everyone. I'm Michael Januszewski. I'm a research scientist at Google. My formal background is in physics, actually, not neuroscience. But what keeps me busy these days is trying to build software to automate connectome mapping. So the goal is to make the data analysis a non-issue. It would be great if you could acquire whatever data set of brain you want and have it basically automatically analyzed so that you can do science with it rather than spend your time and money on stuff like proofreading or stitching or segmentation. So that's what we are trying to do, and to do this automatically.
Hey everyone, my name is Dorkenwald. My work is directly adjacent to Mia's, trying to reconstruct connectomes and solving other problems other than the automated reconstruction. My work has contributed to like the 5-hour project and the Microns project that you just read about.
Hi, I'm Helene Schmidt, and I'm from Frankfurt, Ernst Ringmann Institute in Germany. And my lab is working on connectomics of mammalian navigation, and our aim is to acquire large-scale 3D dataset of the full entorhinal cortex and hippocampus circuit.
Hi, everyone. I'm Andrew Payne, co-founder and CEO at E11 Bio, which is a nonprofit focused research organization. We are a young org that is building tools for optical connectomics to try to make that field move faster now that this technology is ready for extracting molecular information alongside the static connectome.
Thanks, everybody. My question for you is, what would it take to decode a non-trivial memory from a static connectome? No, actually, so one way to approach this is to ask each of you, given your expertise and what you do on a day-to-day basis, if there is a specific advance or obstacle that, if you solved that obstacle, would you believe increase the efficiency with which that path could be accessed to decode a non-trivial memory? Will you talk about what that even means as well? Andrew, do you want to hazard an answer?
Yeah, I mean, I think I'll just start by saying that there are a couple of new, pieces on the board that everyone should become familiar with. The first is that it is now possible to extract connectomic information using conventional light microscopes. And so everyone saw Tabacoli et al. 2025 on the screen. That preprint, definitely check it out. It is the first, bona fide demonstration of extracting connectivity on a cheap light microscope. Why is that important? It's not just that it's easier to get the information, but it means you can use multiple colors. And when you have multiple colors, you can start thinking about what other molecular details can you co-detect with your connectome easily in order to more accessibly decode that memory. For example, there is like a whole body of literature on tagging CFOS cells. That's easy to see if you can get the molecular information in the same assay. And so at E11, we just dropped a preprint last month demonstrating how you can read out 24 different protein molecules in the same sample when getting this kind of morphological reconstruction of the neurons. This could be a game changer, and I'm going to position those two pieces on the board. Maybe they'll come up again as we keep talking.
I would also just encourage the panelists to argue amongst yourselves as much as possible.
Well, we can argue about what the best way is to view connectomics. We may do that tonight. I think even if you assume we have a whole mouse brain connectome, I would hazard a guess that we have a problem or a challenge to read out any memories from it a priori. And I think maybe that is the frame we can think about the scope one moment. Like even if you have the whole connectome, can you read out a memory from it? And I think from working in the fruit fly, one thing that really stuck is that in the fruit fly, a lot of connectomics work really benefits from having the sensory modalities being so close to the brain and kind of understanding what they relate to, the behavior assays that are being done and how they can be related to the connectome. That is a very, very short path to associate those. And I think that allows people to do interesting neuroscience and potentially read out memories. So I think we ultimately need to understand what it means to read out a memory. And I think that has to be related to behavior or to a function of those neurons. So we will need that information to have a chance to read that out. And I do like the Songbird. That's the first system that I really got to work with. I think the Songbird song is a wonderful modality, and the hypotheses are very, very clear. And I think that is one of the lowest hanging fruits that we have to read out a memory and seeing if there is actually a chain of synapses and neurons in the HPC. I would guess there's not, and then we have to answer the question of what we do next and how we kind of go about reading that out.
Well, I think before we go and can read out a memory, I think there is a step before that, so that we need to narrow down the search space. What connections are we looking at? What areas where, like, just to narrow down the space, right? And then we can think about experiments.
I'm gonna do a yes and on what Helene and Sam are saying. I think they're both right. I think what you want to do if you want to read out a memory is to have a learned function which maps some inputs to some outputs. Ideally, you will have the inputs and outputs quantifiable in terms of information theory. So, should be able to count bits that makes the experiment scalable. But so yes, you want to be close to some sort of sensory input, or you want to be able to map your sensory input in the area that you're scanning. I don't think you need the whole brain for this. I think we know enough, from existing neuroscience projects to be able to target specific brain areas, like maybe, the lateral amygdala for fear learning, and other like HVC and RA in the Songbird and so on. It's going to be expensive because of the data analysis challenges, but that's getting cheaper. I think it's mostly a matter of organization and maybe some microscopy challenges, which I'm not so clear on, but we can also discuss later.
So for the Songbird itself, right, what would be the right experiment? I would imagine that something that would be more impressive would be to decode different songs from different birds. Would that be the right experiment? What would be the right experiment?
Just to respond directly to the question, different birds, different songs, I personally don't find that as interesting, because if you want to decode a non-trivial memory, then one of the things you want to do is to do more than just identify this animal has learned something, and here we can find what it has learned. But you want to be able to differentiate between different memories. This is where you start talking about things like how many bits of memory do we want to be able to return? And that's where it would be very interesting if you can explore and find multiple songs in one bird, for instance.
So, I'm not a third neuroscientist, but if I understand correctly, for at least zebra finches, for instance, is one song per bird, right? Is it good enough or not? Wrong organism. Okay. I mean, for what it's worth, to stick to the zebra finch example, I think it actually is a good model, and it would be interesting even to only, for a particular bird to be able to say, oh, the song that it learned, is composed of 4 syllables, and the length of the syllables is that and that, and maybe you also can get some frequency information about those syllables. Already that would be interesting, and it's also quantifiable in terms of, bits or whatever you want to use. So, I mean, I don't think it's trivial at all.
This is where I get confused about the difference between a behavior and a memory, because it was an alternate proposition was if you could accurately decode the last 10 bits of behavior of an organism before the sacrificing of the organism to fix its brain. So how do you guys think about the difference between, because a song is in some sense a memory because you have to have learned it, but it's also a behavior. So are those meaningful differences between a memory and a behavior? What do you think would be a more tractable, the last 10 bits of behavior or a non-trivial memory to decode? Which of those two are we closer to?
At the risk of saying things about songbirds that I'm not aware of. So I think when you're talking about a behavior, the behavior, of course, is composed of a number of different steps, for instance, in that sequence of a song. So then you would say, okay, if I can differentiate between different pitches, different interval lengths, things like that, then you're decoding something significant about the behavior that requires learning. Therefore, there is memory of some sort, right? Because memory, in essence, is anything where, future is dependent on past. And yeah, so to me, that would be a memory as well, or it would be learned memory if you decompose it into those parts of the behavior.
There is, so I will risk this. There seems to be a rise in attention to the cognitive functions of astrocytes, so that it's not just all in the neurons and the synapses. Do you guys see astrocytes as being, or other glia, for example, as being important in these kinds of issues, or is the connectome enough?
I think we have been a little bit reductionist when we talk about what we mean as a connectome. And I think people may think first of the synapses between the neurons, but I really think it is much more. It is glia cells. It is all the ultrastructure that goes along with it. It's the neurotransmitters that are expressed by those synapses and all of that. And we may talk about this as an annotated connectome, but I think most of us don't think about this as a binary connectivity matrix. And I think that's very important. I think the role of glia cells is becoming more and more clear to be fundamental to how the brain works as a reason why we have so many more glia cells in human brains, I believe. So I think, yes, we should not disregard anything of glia cells.
I mean, again, yes, and in the sense that, yeah, the glia are clearly important, but I think, at least in some of the theories of memory that we would want to maybe verify, the prediction is that the memory is stored in, axon network and synapses. So then we would ideally be able to ignore the glia. Although I guess we'll find out once in time. Nobody else.
I think I'll bite on the temporal component. When we think about recent work, like for example from Adam Cohen and colleagues on the epsilon like pulse chase dyes, we know that we can measure in a molecular assay, synapse turnover and synapse plasticity, and we can just around the corner, a fearless prediction, we'll be able to detect that information. What is the T-1 connectome? What is the T0 connectome? And depending on whether we can develop more halo tag like dyes, the T+1. So we can get a couple of different time points. And that gives you information about, what neurons you could push on if you were simulating the connectome that you have observed. But the question is, what model would you use where you could actually use that information quickly? And maybe Sven has a thought on that.
Yeah, so the temporal aspect, right? So the goal is to decode from a static connectome, but as soon as you start talking about temporality, I mean, I guess it's still technically
slices of static connectome in that sense, but you add the temporal aspect to it.
I think it still counts as a static connectome. You're reading it out in a single time point in a single asset.
So are there other obstacles that you face in your day-to-day work that you think would solving those obstacles? Or sorry, but
I think challenges that we are facing are very clearly that our data is extremely noisy in the end. I think the work in Drosophila is showing, and the first work really to compare connectomes is very important, and it's showing that there are many variability, sources of variability that we are facing. And one of them is technical noise. And we may only be able to suppress technical noise due to errors, imaging, artifacts, and all that to a certain degree. I think it's a question if that is a sufficiently low degree that we can push that. I believe so. But there are other sources of variability, biological variability between individuals. Ultimately, we have to define that first before I think we can understand what is real differences and what may constitute real differences then between learned memories between different brains. And to that also belongs accuracy of synapse readout. And I think our work may have contributed to that a little bit, that maybe you only have to read out if a synapse is large or small. Maybe that is sufficient. I think more work needs to be done. But that would certainly be helpful to knowing if that is the level of accuracy that's really needed. I think other labs have done separate work on that in hippocampus, showing that we may need to read out synapses at a level of 10 or 14 bits of accuracy.
so you think the variability itself, it's an open question whether the variability or the stochasticity is an obstacle, but because it's an inherent feature of the system.
Well, I see actually a difference between those two brains, or if it's just like happenstance of noise that happens naturally. Like this one brain was acquired in the morning, the other one in the afternoon. Is that enough that creates those differences? Like, I think we have to get a handle on this first. So far, Connectomes has been a field of N of 1, and we are slowly getting to a place where we have an N of 2, 3, 4 in Drosophila. I think we really need that too in the mouse to understand differences between brains.
So, fine question to you. Let's say we don't have this perfect understanding of variability, and by the way, memory is a source of variability, right, between specimens. So let's say we don't have the perfect handle and understanding on everything, but you can show that you can make predictions about what the organism actually learned, meaning that you look at the connectome and you make some correlations about the inputs that were, associated with whatever, some positive or negative feedback. Would that work in your mind or not?
Are you saying as long as you have a model that can that you can show that you can read out a difference in behavior from a system, is that enough?
Yeah, okay. I'm asking you because you said, we have to understand everything first. You have to understand social variability. I'm saying, well, we don't understand it, but you show the correlations, and the correlations, are statistically significant. Is what you know.
How do you show statistical significance in this? I think that is the point, right? That you need that error bar. What is statistic?
Well, okay, so you can approach it from like almost kind of statistical theory, right? And we're getting into hard experiments here, but let's say, you have an animal, it has some input, maybe it's like, tones or colors or whatever, and you learn, you have each individual learn some behaviors, right? So maybe the bad combination, like combination of A and C is bad, A and D is bad, A and B is good, B and D is good, whatever. Like you can include some number of those things in some brain area. You take the connectome, and then you look at the structure, and you make a prediction, right? So somebody does the analysis, and you say A and B good, A and C bad, and so on. Some of us is going to be correct, some of us is going to be incorrect. If you say, okay, but it's only N of 1, then you do more samples and more animals, and you can keep doing this, right? At some point, you will reach statistical significance, however you want to define it. And you will not understand necessarily all the sources of variability, but you will have a statistically significant correlation. Is that good enough? That's an open question, right? How do you define this?
I want to leave that to other panelists. I think it's. What do you find convincing?
No response. Sure. We can take questions.
Sorry, I used my privilege. So I think when I think about these things, I think that the very first question is we have models of how these systems work. These are model organisms that have had decades of research on them. And so the very first thing that you're doing is you're saying, here is our model of the hippocampus. Here is our model of how place cells and grid cells interact with each other. Here is our model of how a place cell gets formed in a single instance. Now, given that model, which could be wrong, what is the experiment that we could do to try to decode the memory based on that model to show that it's wrong or to show that it's right? In terms of the bird song, the bird song is very concrete. This is not like, oh, yes, we could decode a bird song. I want people to understand that, right? There is a theory, like you said, Stet, that could be completely wrong, right? That there is a chain of neurons in HPC that sequentially activate like a timing board. And that those chain of synaptic, of neuronal activations are then feeding their axons down to a motor nucleus that gives particular tweets. And the textbook model says that if that theory is correct, then this connectome will show what the bird learned. I think that this is the kind of concrete thing that I'm hoping that we can discuss. We know that there are dozens of model organisms that have decades of research, and connectomes can get those and decode memories, or fail to, and show that those are.
So because you were talking about place cells and grid cells.
Please give us a tutorial.
No, I would just propose an experiment, like teaching a mouse like a parkour, like first go on the bridge, then to the stairs, then to whatever treadmill, and have several stations like this, and then whatever, take 10 or 20 mice, and then they have to learn each of them a different sequence. And then we have a model. Well, we need to figure out first where to look at, right? Exactly what connections to look at. And then we would try to predict then what sequence was learned.
I think for the start, I would start with a very simple T-maze, go left, right, and see if I can read that out of the connectome, right? I mean, it's not 10 bits. It is not 10 bits. But I think that would constitute for reading out of memories left. I think I'm enamored by hippocampal replay and that we see in sleep. I think there's evidence that there's something in the connectivity between those neurons that we should be able to read out, and that is certainly something I think that would be worth targeting.
But so just going back to the structure versus function problem, is it an insurmountable problem that, for example, in artificial networks, you can freeze the weights and generate different dynamics, even with a static frozen weights within the network? And so there's this problem of multiple realizability and degeneracy in the dynamics of neural activity. Is that not a fundamental hurdle? Or is there a way, do we believe that we can, in some sense, analyzing the structure, have a measure of some capacity of possible functions, for example? Or is it a fundamental hurdle?
I mean, to that point, I think what we're missing is a lot of annotations in the connectome, including the biophysics of those neurons, which I think will lead to multiple possible outcomes when you model such a circuit. So yes, I think we have some of those problems. A connectome alone may not allow us to encode that. However, I think work by Philip Xu in the Drosophila connectome kind of showed that even with a model that is not perfect, you are able to produce activity that matches measured outcomes. And I think that's very encouraging to see.
I think we are being too cautious in this discussion. We are finding potential problems before we have even started or designed an experiment, right? Like maybe we don't know sources of variability. Maybe we don't have the biomolecular information. Maybe there's degeneracy. There's an endless source of potential problems. But why not just try? And maybe we fail. In which case, of course, you have to go back to the drawing board and start thinking about those problems. Maybe it just works. In Drosophila, it actually kind of does work, right? Not the memory reading, but reproducing some stereotypical behavior from, not fully perfect connectome and with a lot of missing information, but yet it was still, a kind of strong enough behavior, but it was relatively easy to reproduce. Maybe strongly encoded memory is just like that. Why not try?
Feel free also to come up to the microphone to ask questions. Anyone I can hand the microphone out, or you can come up to this microphone in the front as well.
I have a question.
Oh, sorry.
It's all right. In what year do you expect that we'll see the first decoding of a non-trivial memory from Connectome?
1987. I think this is a good hazard a guess, please, everyone.
I think this would happen once we can reconstruct the whole HVC region, which is, I think, possible now. So I would say within the next 5 years, we should be able to see that.
I would say the same within the next couple of years, provided we actually start doing this. I'm talking about
it. Randall, you have to guess.
Okay, I'll guess. I'm just agreeing. I think that, yes, the whole point is to just get started. I thought it was wonderful to just say, why not try? So, yeah, 2 to 5 years is my guess. I'm going to be really optimistic.
Miranda?
I agree with the 5. I go with the 5 years.
Good God.
I mean, okay, I'm going to ask the question that I think needs to be asked, which is, nontrivial is doing a lot of work here. What does nontrivial mean? And actually, I would love if some folks, came up to the microphone and offered their perspective on what does nontrivial look like. For example, we know in like the retinotectic system that you can sensitize that to a moving bar, and you can read out the like change in like the compound synaptic currents from that, and probably if you had the connectome, you could see that in that system. Have we already decoded that memory, or is that too trivial? It probably is, but what is the threshold? Can we get some discussion around that? Also 5 years.
This, by the way, is why I think we should have this quantified in like information period terms, so number of bits, and then we can argue about the number of bits by this one trivial. It's become a threshold problem.
I feel like I could make a lot of money from you guys if we just place a wager about this, because my guess is I'm much more pessimistic than all of you. I mean, it's awesome, and why not try? And yes, rainbows and all that great stuff. But I would guess 30 plus years.
Jesus. Please say, we even have a whole mouse brain connectome before then.
Yeah, like it was mentioned, non-trivial is doing a lot of work, and I think that I don't know semantically what that even really means.
I would also say no rainbows needed. We focus on do EM versus grayscale, no rainbows.
Right.
It's probably just a comment. Just trying to think about what a memory is. Is a memory indeed just written in the weights of synapses and the structure, or does memory needs to be experienced? Like, do you have to feel? Do you have to live through the memory? I'm just talking about this, thinking about astrocytes. If we know astrocytes modulate the synapse. So what is the substrate? So if this is true, then your synapse sizes and weights and whatnot is not enough. Then you need to have the substrate of the astrocytes and how they influence. And the question is, are we going to find the substrate of how or whether this influence is encoded in the structure of the astrocyte in any way? Whether we can actually see it in the structure, and is it necessary for the experience of the memory? Yeah, is a memory experience, or is it just something written down in book? That's something to think about.
Anyone want to comment on the nature of memory?
Yeah, so obviously there are a lot of details you could get into about what constitutes memory, what contributes to memory, what modulates memory. But again, you could probably start by just counting synapses, or looking at the size of dendritic spines, or looking at postsynaptic densities, because that's enough to begin to look at least some memory and to identify a non-trivial set of different memories. Think, for example, receptive fields. We know that we can identify receptive fields just by looking at synapses without taking astrocytes into account. And then if you combine receptive fields into feature detectors, you can see that as well, just looking at synapses. So I believe there's enough there that you can decode something even if you can't decode everything, even if later on you discover there's more you need to know.
Does anyone else want to comment before we move on to another question? Okay.
So wanted to come back to understand a little bit more what people want to focus on memories, because there are so many types of memories we have discussed. There are episodic memories where one can try to go and say, hey, I actually remember walking in this room or walking towards the microphone and saying something which is incredibly difficult, might be much closer to 30 years, but there are going to be memories which are going to be, which are fundamentally on how the system is built. And from that perspective, I wanted to bring a very concrete question. So let's say that there was an element to say that if we can predict, for example, the Saki field. So if one goes into right now into microns, and we have the capacity to predict how the neurons are going to respond to stimuli without looking at how the neurons are responding, just looking at their connectome. And let's put a fraction of an explainable variance. What would be a fraction of explainable variance which would satisfy the equilibrium? 30%? 10%? Neurons are quite variable, by the way. Very concretely. What would the committee say from here? What would be the fraction of explained variance that would quantifiably say 1 trillion or more?
We are not a committee. We're just a bunch of people. And in the end, we don't decide this. Maybe we should do a vote in the audience. Let's see what people think. No, really, because but here's one thing. When we do the memory readout, ultimately, this is going to be judged not by the 5 people here, and not even by the people in the room, but by the wider audience, right? And by that mean both the lay audience and other scientists and so on. And whatever we come up with, it has to be convincing to that group of people, basically. Yeah.
I just want to say that is exactly the right question. Okay, that is exactly the right question, and it does not make it trivial that it's like, oh, you're just trying to explain a certain amount of variance. It's like, we reported from 10,000 cells, calcium imaging, and we have the connectome, and how much of that calcium imaging could be predicted based upon the connectome. If we are right about the theories, about how structure encodes function, then that experiment will get a high level of predictability. If we are wrong, which we may very well be, then it won't. It tells us something that other experiments have not told us. And that's the whole key to this non-trivial. The word non-trivial is moving forward the neuroscience of how structure gives rise to function. That's really, I don't know what the criteria, what the level would be. That is something for like a committee to decide. And it is something for to say, this is where we are. We're not going to decode a memory of a childhood experience of a human. Okay. And we've already decoded to a certain extent receptive fields of single neurons. Really puts our theories to the test. And that's what non-trivial means, to me at least. Although I'm super curious about other people's.
Just to let you know that Fiti does a relatively simple linear network to microns data, gets you slightly below 20% of the explained variance. So the type of theories we should expect, what would be kind of like the standard model of artificial neuronal networks, doesn't get you that as much as what you'd expect. That's at least in our hands. Maybe somebody could read that.
Yeah, so I have another question for the committee and for the audience. So when I think of a non-trivial memory, I think of something that I experienced, right? And you think about how neurons were responding in my brain. You need to think about what the neurons represent. And so when I relive that memory, we think the neurons that were active when I was experiencing it are sort of replaying it for me. So in that case, and I'm glad we're talking about microns, because microns is a data set where we have function and structure, and that's like really critical to me when I think about memory, right? Because a non-trivial memory kind of requires that we know what neurons represent. And so is it cheating then to measure the function first? Because for me, I can't really see a way that we could decode from the connectome if we don't have a sense of what each neuron represents in the living brain.
I 100% agree. And I think that is partly to my point from earlier with Drosophila connectomics, where you can get away from having functional experiments because you can associate neurons much better with behavior. I think in lieu of that, you need functional experiments so you can actually associate those neurons with a memory, with a behavior. So I think drifting gradings is a readout that is certainly convincing. For me, memory is much more similar to what a hippocampal experience looks like, because we know it is replayed in sleep. I think that, to me, puts it to a point where I believe that this is actually something that constitutes much more like a convincing memory. If I would tell that to my friends, they would kind of see that and think that this constitutes memory, that we can decode what a neuron responds to a moving bar. That seems to be not to pass the dinner test.
Hi, I'm also very interested in the topic of memory versus behavior. As you probably know, there's the recent critique on LM and AI can't be conscious. There's an argument that because they're just predicting machine reacting of stimuli of input text and predicting output text. But then what is human but also predict machines that reacts on more complex inputs and produce more complex output than text? And in that sense, perhaps memory would just be behavior in patternized. And I guess another side of that question is, what do you think would be the role of artificial neural network in trying to uncover the mechanism of organic memory, and vice versa? What would be the role of uncovering organic memory in developing better?
Be brave and just try. Just try.
I can comment on the experience. I have maybe a strong argument, but we can think about it, and then you guys can tell me if this makes sense or not. Imagine the zebra finch example, right? So in the extreme, you could imagine a system which is basically what I would call EM to MP3. You input a big EM volume of the whole song pathway, but is big enough that it allows you to predict the sequence of the HVC axons. It's deep enough that you see the connections from the array to the muscles, so you can map actually individual axons to frequencies and so on. So you literally input into a big computer program. It runs many GPUs. At the end, you get an MP3. You play the MP3, same as the birds, right? Great, it works. But there is no birds to experience it. Is that a problem? Probably not, right? We did decode the song. So I would say, the experience is not actually needed. What is needed is the information we are extracting from the system. And I can try to tackle the AI question, but you have to repeat it because, unfortunately, can you say again briefly what was the AI question?
Because AI and brains are both neural networks, and you could make a pretty big leap in assuming that there could also how AI encode memory is similar to organic brains. Huge leap, I don't know, but could there be link in uncovering one would help the advance of discovering the other? Something like that.
I guess in some sense there is a analogy. It would lead to current theories of memory, right? If you assume that the memories are encoded at the synapses between axons and dendrites, then those correspond in some sense to the weights in the neural network. So it's not changing the weights; it's kind of like changing the memory of the network. But that's a very high level analogy. I'm not sure if that's if that's what you're looking for.
So I want to try again to put you on the spot, Helena. And this is coming more from my ignorance. Whereas I understand, or at least I think I understand, the HVC to RA songbird system, and it's pretty straightforward. And I think I understand the visual system where I say, the receptive field of this cell is built up by the sum of the receptive fields of the cells that come before it, with maybe some lateral connections. What I'd really love to hear is if I'm a mouse that has learned a path. Now that's a good solid memory that people can kind of get to wrap their heads around, even better than a bird song. Do we have a theory of how that occurs? I would assume we have several. And given that theory, is a decoding of a learned path, location, T maze, as you were saying, can you imagine an experiment in the next 5 years that could decode a path that was learned in a mouse?
So I think my approach to this would be the thing is we don't know. I think this is the answer. That's why we are going to get the whole connectome, right, to see the connections and so on. There are so many models, obviously.
You're about to say, I mean, we don't know, but we have a bunch of models that could be right and could be wrong, or we just don't have models.
No, there's a bunch of models. For example, the feeder model that has like precise connectivity predictions between grid and place cells that are fixed, non-random or random, and a pathway that is learned where they expect where the learning is happening, happy learning or plasticity. But we need the connectome to see if it really is the case.
So, I mean, could you explain to me, and I think other people are interested in this, naively, how could learning a path, learning a spatial arrangement of this room be encoded in synapses? I think as neuroscientists, we that are really deeply involved in these models, we take for granted that that's obvious. It is anything but obvious to most people, including myself. Is it too complicated to explain? This is how this memory of a spatial path is encoded in these neurons, in these synapses.
Well, you would expect some sequences, right? Of neuronal sequences that you would probably see, but to be honest, we don't know what connections to look at exactly.
CA3 and CA1. And,
well, my guess would be CA1, yes, but still, connectivity is really low in CA1, so yeah.
Okay. Yes.
Wow. Hi everyone. So I wanted to ask you about something that was I requested in the beginning, which is to understand what each of you think is the biggest bottleneck in getting to the next level. And what I was surprised about is I didn't hear any of you mentioning proofreading. Right? And like when I have to spend the money, it's still like $1,000 per neuron the last time I checked. So I'm curious on different takes on that, or maybe you like all uniformly think that that's all solved problems. What would the case be?
I think I'm the last person you find that would say that this is a solved problem. I think, though, it's interesting to think, what if we don't have to think about proofreading for a moment and what we can do without that. But I think you're right that manual proofreading to this day is the main bottleneck. We have recorded over 10 million manual edits across many data sets, including the Flybrain, the recent bank data set. Microns has seen 1.5 million manual edits. You can just imagine how much manual work that was done, and it's not even reconstructed the entire data set. We are at a 2% also of the entire data set. So this is clearly the main bottleneck. But I think between me and maybe disagree a little bit how far along we are at this path, though I still think that many of us are working towards this goal of reducing the need for proofreading, and I think we're making big progress. I think the irony is right now that we have been scaling our data sets faster than we've improving our methods. And the consequence of that is that we need to do more proofreading today than we have in the past, simply because our data sets are larger and require more manual labor.
I'm actually not going to disagree with this. What Stan said is, of course, correct. And the problem that Anton brought up is it's a bottleneck. Proofreading is costly and takes a lot of money. But what we have to keep in mind, and this is, again, to not be too pessimistic, you have to keep in mind that the data sets that you see published today, those are data sets that were acquired maybe 5 years ago and have been processed over that time. Better technology exists today. If we do the memory reading or any other experiment today, this is most likely going to be done with this better technology where the proofreading cost can be 2 orders of magnitude lower.
And that's a big difference. My question is sort of drilling down into what non-trivial memory is. So the simplest experiment that I could think of is in a fruit fly, you give the fly some sugar on its legs, it will typically extend its proboscis. You can punish the fly by giving the fly sugar on its leg and, in fact, presenting the fly with a bitter substance, and it will learn no longer to extend its proboscis to sugar. This is something like 4 or 5 synapses. The sensory circuit is 5 synapses. There's 20 defined neurons that we have genetic access to, and we could do this experiment, I would guess, in 3 months and find the structural basis of this particular behavior. So my question sort of is, like, would this be a non-trivial behavior? And if not, are there any non-trivial behaviors in flies at all?
I think that is a great point, of course, but you can argue that any olfactory memory that a fly has built is already then rising to that level. And we have shown we can read out a lot of that. And so maybe we already achieved that by the work in the mushroom body that has been done. I'm not sure it rises to, what is a non-trivial memory? I think is the question. I think it comes back to the point of, is this something that can convince your friends at a dinner table? And I'm not sure that making this argument to those would pass that test. That said, I think you can totally make the point. You can read it out of the connectome. You can see the changes, and that is certainly decoding.
I have a question kind of about the definition of memory in a sense, but in comparison to what's the difference between a memory and learning? So, memories are, like has been pointed out earlier, can be episodic, or memories can also reflect as learned behavior to like skilled behavior. So, just as kind of examples, right? Probably everyone remembers their first like conference rejection or like a reviewer 2 comments specifically, has a vivid memory of that. But it's not a learned behavior. But everyone, people who know how to ride a bike, for example, they don't think about that as a memory. So when you decode these 2 things from a connectome, would that be fundamentally different? How do you think about memory versus learned skilled behaviors?
I don't have a good answer to that, but I think it's an excellent point. Yes, I'm sorry, Dennis, I can give you more. Yes.
Thanks.
No, but I think, of course, in the simplest way is that Jennifer Aniston neuron that we can pull out, that we can point to, and that relates to memory. I think for us, we can't look into the fly's or the mouse's head to see what it's thinking in that moment when it sees activity. I think ultimately that is part of the problem here, that we may not know what the mouse experiences in that moment.
Sorry, not going to go easy on you since I have a similar question, and I think a lot of us maybe do. But it's sort of a 2-part question about going back into how you maybe would define a memory, as well as thinking about multiple modalities for memory and how you might explore them. But coming from a cerebellar background, I know thinking about things like vestibulo-ocular reflex, which is a reflex, but you can sort of train to adapt to sort of skew that reflex. Would you consider on like more of a molecular scale, could that be considered a memory, the sort of training that needs to be done? And then my other question is, how would you maybe think about designing experiments that are not just episodic, but other kinds of procedural, like a Proustian memory in the olfactory system? Would you design experiments differently to try to, or would you think about evaluating the data differently to decode those different types of memories?
These are all very good questions. Okay, we'll keep that one in mind. You guys crunch on those questions, and maybe you'll come to an answer. Go ahead.
I think I'm last in line here. So going back to, so there have been several structural features that we sort of were concentrating on the readout of the non-trivial part, but the left for a moment the structural parts. So there have been several structural parts that have been mentioned, and the synapses, the strength of them, maybe the size, the length of dendrites, or the neurites, whatever. And I admire the optimist that let's just go for it and try if the reading those structural features is enough. But if it isn't for mammalian cell, what would be your next bet? Like, what other structural feature do you think would be like the next thing that might need to be concentrated, like plan B, like if in case it doesn't work for mammalian systems specifically?
Yeah, I mean, just to throw something out there, I will bring us back to the epsilon system with the pulse chase dye. The reason for that is because you can figure out exactly which synapses participated in a memory formation. This work from Adam Cohen was published in Nature Neuroscience earlier this year. Definitely check it out. So there is, I think, a lot of implicit value in knowing, okay, which specific synapses participated. That doesn't decode the memory, but it gives you a way to like narrow in much more quickly on which parts of the representation are important.
I think trying and failing is a very important step in this to see if we can do with the synapses and the connectivity that we have right now. I am personally extremely excited by the foresight of having molecular annotated connectomes that Lycons can bring us. And I think having annotations of neurotransmitters especially has been proven indispensable in flight connectomes. So I hope that adding these kind of labels, molecular labels, will bring us closer to being able to better model connectomes in mammalian systems.
Nobody here thinks that neuromodulators are an important factor?
If we have no idea how to deal with them right now.
I mean, I think what you're trying to get to is that there wasn't a necessity for the size of a dendritic spine synapse or the size of the PSD to be well correlated with its function. Now people have shown this. Haller et al. Showed this beautifully that it was in this particular type. But for instance, in the mushroom body, it's not immediately clear, at least to me, that there is a structural synapse signature of a memory, and yet we know that there's memory. But what we do know is that there is some physical mechanism that activates these neurons. And so the next level down, I think your question, and I think Sven, you were saying this, is the molecules, the neurotransmitters, but also the receptors. If you're counting, and I think Andrew, you were saying this, if you're counting the AMPA receptors that have been inserted into a spine, that's got to be the true functional connection, because those are the things that are opening up that give the current that connects one neuron to another, even if the structural size of that synapse is uncorrelated.
Another question?
Yeah, so we saw in the Spires-Jones presidential lecture on Alzheimer's last night, 2 nights ago, that even with 4% synapse loss, we were seeing pretty catastrophic issues with memory and maybe near fatal pathologies, 4%, right? So that kind of gives us an accuracy envelope that we need to fit inside of, if we assume that that's like causally linked to the memory loss, right? So do you think that we're on track now? Maybe there now or on track now to kind of fit within that, accurate plus or minus, that last 4% to get maybe catastrophic memory loss reconstructions, and then marching forward to accurate reconstructions where we're maybe conserving those memories and synaptic reconstructions?
I have a question about the lecture, which I unfortunately did not catch. Was there any information on the synapses being, the 4% missing? Was it random or correlated? Those specific cells?
Probably not random. I mean, I agree. There's like this possibility that memory is like holographic, right? And like kind of diffused across the brain. And then there's of course this notion that like memory is probably not holographic and it's not like uniformly distributed, right? And it's probably halfway between.
I think 4% is an extremely low bar, and I would be very much worried that we don't pass that bar right now. And I think one of the problems is that we don't know if we have passed this bar, because Connectomes is treated as the ground truth right now. Testing to itself is hard, so I am very much worried about the 4% bar.
Andrew, it looked like were you in that?
Yes.
What he said.
Okay. I would like to venture an answer to one of Ken's question, and I have a question to ask. This should be easy to answer for the committee, but I'll venture my answer first. Okay. I'm going to propose a result that I think will make the decoding of the place cell memory. Okay. Let's suppose I have an animal. I record a bunch of place cells. Let's say I just record 10, and I know their place field. And then I do a connectomes of this place cell. I reconstruct all the connectivity. And I think the test would be that if you tell me only the place field of one of those place cells, and I can predict the place fields of the other 9. And if those are right, and I would say that's a non-trivial decoding of the spatial, just a minimally variable spatial memory, because I can decode the other 9. Meaning that, and from the relationship from the connectivity, I can decode, the room basically. So that's my, my 2 cents to your question.
Can I ask what regions? This is a hippocampal expert in connectomics. What regions of the hippocampus do you think you would have to have connectivity for to pull that off?
I would give a minimally viable answer, and I would give a more, more complicated answer or whatever. Minimally viable, I think it's probably some part of CA3 that have enough numbers of place cell. But I actually don't think that's going to be sufficient, because these neurons projected to contralateral hemisphere, who knows that these kind of place cells and the prediction that I'm calling is actually going to the other side. So it might well need the whole hippocampus. And hippocampus happen to be really big and, has this weird banana shape that span pretty much the whole brain. So I guess we're talking about recording a bunch of place cells and pretty much reconstruct almost half, if not the whole brain. But it could be done. Excuse me. It could be done in one animal, though. I think it's possible. Okay, I have a totally unrelated question. If you have to say, which model organism is going to be the first to be decoded, a non-trivial memory, and you can only give one answer, whatever your definition of non-trivial memory, what model organism, you would vote for?
Just to echo Sven, the mouse is going to pass the dinner table test, and maybe it's just like knowing left or right, but that I think might classify as the solution. But
well, I would go for the trust control, but it's hard to do.
I think the songbird is closest at this point, and I think it has the most well-established example of what would constitute a readout.
I feel like some controversy is needed here, so just something that. All right, and I'll add zebrafish, because we shouldn't forget about the fish, and because it's so nice and easy to ground or validate what you believe that you found using calcium imaging. So
I will go pterodactyl.
So I wonder if there was a year in genetics that was kind of like where neuroscience is at, maybe 1970, where you hadn't had a human genome project. You knew there were base pairs and all that stuff. And so I wonder, the question is, to really understand how brain works, are we just going to have to decode some human brains and just have full connectomes? Because otherwise, I guess that's my question. Can you really understand how human brains work until you do that? And if so, is it inevitable that we'll have to decode some human brains so we have that. Because how else can you get this full knowledge without doing that?
I think there will be, like with the genome, there will be at base, a whole brain. Maybe it's a conglomerate of multiple ones that kind of leads as the anchor. And then you start comparing to that, and you will not acquire a whole new one after that. Once you understand what regions are relevant to your question, you may start just doing those regions and comparing and comparing and comparing. I think that's what we can learn from genomics. However, I think brains, while we have parallels between connectomics and genomics, there's huge differences. And I think the brain is, in the genome, you know somewhat what your entities are. We talk about, is it the synapse? Is it the size of the synapse? Does it have two states? Is it a molecular level? Like, what is our fundamental level that we actually have to understand to start comparing? So I think at some point the comparison to genomics will break down for connectomics.
Yeah, and just to echo Ken, if the thing that you're interested in is testing a hypothesis around how grid and place cells encode information, like, we don't need to map the human brain in order to start testing that hypothesis when we have the connectome of the hippocampus, of the mouse hippocampus, or the tree.
Does it matter that a memory is a process? It's not a thing, that memories change. When you actuate a memory, you're actually changing it. By reliving it, you do change it until the fish is bigger, etc.
So that's not always the case. For the zebra-finch, just dwell on this. The song is learned once for life. It doesn't change. It's crystallized.
Sorry, what is learned once?
The song of the zebra-finch. Mm-hmm. So, I mean, it's a specific example, but this one where...
I mean, would that make it trivial then, perhaps?
You guys tell me, but there's the EM to MP3 example. It's not trivial. You can literally play back the song. I think it was very hard, but in principle, possible. I don't think it's trivial.
Just because you asked process versus static, and it seems to me that for the decoding prize, at least, the process doesn't necessarily matter. It's very interesting to know how learning establishes memory and how memory changes over time. But for the decoding challenge itself, that shouldn't really be a hindrance.
So maybe we're not decoding memory the way I understand memory, but memory the way a decoder understands memory or something.
Kind of a naive question that's maybe a little different from the structural connectome, but about maybe functional connectomes. So if you have a clear zebrafish and you could do maybe holographic stimulation of every neuron, every pair of neurons, and see what lights up, is that going to be enough to decode memories? And would some combination of structural and functional like that be enough to do anything useful?
I think to qualify, we need the structure, because ultimately I think the question is, given the structure, a snapshot of it, can you read out a memory? And so I think that is needed. But I think having all the activity for all the neurons and is certainly something that will help us, I think, get at that question. So I think also, to your point, a process and a memory, I think we look at a snapshot of a brain in the end. So we look at a snapshot of memory just in the same way. The memory might have been like, your taste of spaghettis might have changed over time, but we asked the question at the point when your brain was looked at, what did you think about spaghettis? And I think that is what I think we talk about when we talk about your memory that we want to decode.
But then you never actually have a memory if every memory is an instant in time, right?
A memory is an instant. I would argue a memory is an instant. And memories can change.
Right.
Sure.
Yeah.
I agree.
So I've just been wondering about how much of the problem of decoding a memory is actually going from structure to a simulation. So, for example, if you think about the songbird, if you want to go to an MP3, you have to basically simulate how these neurons interact and what sequence they're active, and then you also have to somehow ground that in how they are connected to the muscles, and then simulate that. So there's a big question of do we have the good enough model to infer the function from the structure? And like for example, if you find strong synaptic connections between certain neurons and you say, okay, that's my memory, but it could be that these are unrelated to the memory. Maybe there are experiments where you can show that these are the synapses that changed, but still,
does this prove that this is storing the memory? I think you have to actually, like if you ask a human, do you remember this or that? Or if you ask an animal, do you remember that? What you do is you have the animal perform some motor action, like the human responds to you, okay, it was like that, or the mouse turns right, or something like that. So you need this model which infers the behavior from the structure. And let's say we assume that synaptic strength encodes the memory, and we simulate it and we find, okay, we didn't get the right answer, so then maybe our model is wrong. And it's still in the synapses, but you have to have a really good model to simulate how the activity results from the connections. Is that the actual lead problem of decoding memory?
I agree with you that that's super important, whether it's a memory representation that we were talking about memory right now or any other, like say the dots of ink on a page of paper don't mean anything without a decoder, right? The retrieval, how you retrieve it determines what it means. But for a decoding experiment like this, these things, while very important, don't need to be in the same experiment. So if you have a system for which you already understand from other experiments, say functional experiments, to some degree how the decoder works, how you can interpret some aspect of what these synapses here may mean, then I believe that you can use a static snapshot to come up with a decoding experiment without having to do that for each of those memories as well. Correlate. At least I hope so.
There are a couple points that caught my attention. One has to do with this idea that once you've got a connectome, you can actually have a place-sum map. My understanding is that's non-paltry, that place-sum maps is remapped based on contents. So given any hippocampus, that only is a connectome in context. So I think, so that's part one. So, that kind of addresses this question of what kind of memory can be addressed or what can be coded without understanding the decoding language to pull out this just happened there. The other question is, what year are we in with respect to genetics? And we're not even close to the settlements. I think that there's one really fundamental question which is not answered, which at an information level has to do with the information parallel is we don't have a language to understand the dynamics. If there happens to be a parsed semantic of neuron dynamics, just the way we have a machine code for computer science, or we've had the code on, we don't understand in behavioral neuroscience right now what the correlate is of what genetics calls proteins. We're beginning to get the idea that there is a DNA. We understand sequences of firearms, but we don't understand the way in which those are contextualized in a separate building blocks for actually what behavior is looked at. And the remapping of hippocampus is a great example. You can have exactly the same codes coming out and then you completely different things based on context. Anyway, so for comment, you have to pick if you wish.
Regarding your first question, I absolutely agree. So those experiments would have to be in a context, and probably one would have to do, one would have to record the activity of a place cell, like some functional recordings, not just static connecting.
So I'm told we are wrapping up the panel. Does anyone have any final thoughts? Has anyone changed their optimism or pessimism in any regard over the past hour? Okay, well, thanks for all the questions, and thanks to the panelists for
Brain Inspired is powered by The Transmitter, an online publication that aims to deliver useful information, insights, and tools to build bridges across neuroscience and advance research. Visit thetransmitter.org to explore the latest neuroscience news and perspectives written by journalists and scientists. If you value Brain Inspired, support it through Patreon to access full-length episodes, join our Discord community, and even influence who I invite to the podcast, go to braininspired.co to learn more. The music you hear is a little slow, jazzy blues performed by my friend, Kyle Donovan. Thank you for your support. See you next time.